I'm going to try and convince you that this is a biosphere that literally touches us all. And so to get started with just some general motivations, why should we study aerobiology? So often a common environmental question that we start with is, how does it affect global biogeochemical cycles? So you're probably pretty aware that there's some carbon in the atmosphere. That's something that's, we've thought a bit about. And so here I'm just putting it in a slightly different perspective. And so here's some global carbon pools all stacked up. And just to make the point that not only is there carbon in the atmosphere, but there's actually more carbon stored in the atmosphere, say, than for all plants on the surface of the Earth. So vast stores of carbon that are of interest. And we also know that microbes can metabolize atmospheric gases. So there's, commonly discussed options such as methane, other sort of trace gases like carbon monoxide, hydrogen, various other volatile organic compounds, moving on up to more complex compounds as well. But the remaining question is, can they do this in situ? Is this a process where microbes are actually actively breathing the atmosphere while they are transiting or residing in that location? And then another way to think about this is by number. So estimates are about 10 to the 24th cells in the atmosphere, which is on the same scale as the number of stars in the universe. So even if they are only metabolizing some of those items on the previous slide at tiny, tiny rates, there's enough of them that this may actually be a very significant part of global carbon cycling that we're currently not accounting for. And then, so I come from the School of Earth and Space Exploration, and so astrobiology is also a very important motivator for a lot of the work that we do at ASU. And so thinking a little bit about understanding the limits to life aspect of this as well. And so here we have a plot that is, or a schematic that's going, from surface habitats, and there's this idea that microbes are going to winnow as you move up to the stratosphere, and then something like the ozone layer may not be transitable and remain viable, at least very easily. And then there's a whole range of different stressors, right? So radiation stress, temperature stress, low pressure stress, water availability stress, nutrient availability stress, and then ozone, as I just mentioned. So the general concept is that the likelihood for survival is going to decrease with increasing altitude, but we do not know that limit. There have been samples that have been returned to Earth with viability up to 41 kilometers in altitude. So things have come from the stratosphere and survived. And then you heard with the poster talk that the ocean is the largest biome, but if we throw the atmosphere in, it's actually slightly larger at 10 to the 10, not that size matters, kilometers cubed. So this actually would then become Earth's largest ecosystem. So we also know that cells have many adaptations to the stressors I just mentioned. So there's lots of examples. So I live out in the desert, right? And so there's many examples of microbes that are capable of living underneath a rock or developing their own sunscreens, creating these EPS layers, various ways to deal with reactive oxygen species, and repair membranes. Various osmoprotectants can help deal with temperature stress and keep their proteins active. And so we know that cells are perfectly capable of doing all of these things with stressors that we've studied well on the surface of the Earth. And then there's always the classic spore formation, just hunker down, get through the transit period, and then activate again once you're back in more hospitable conditions. But there's actually some evidence that not only do cells have these capabilities, but there may be some environments where organisms are actually pre-adapted to be able to handle these stressors. So here's a nice example of two different cultures. So one is this halobacterium that is a mesophilic organism, likes the temperatures and conditions we like, well, a little saltier. And then this Antarctic isolate, also a halobacterium. And so it got a free ride on a stratospheric balloon And then it was compared, so the cultures that remained on the ground versus just any stress that might be induced by being courier transported and then exposed to the stratosphere. And so here's just evidence showing there was more colony forming units after the culture was exposed to the stratosphere from the ones that were from an Antarctic adapted culture as opposed to the ones that were mesophiles. So Antarctic conditions being very cold, also having to deal with some elements of UV and desiccation stress, depending on where you're from, may have provided advantage in this case. So there's also some interesting research that has shown how cells enter the atmosphere may play an important role in survival. And so what I mean by this is, here's a nice experiment where cells were either aerosolized, so bubbles, bubbling a liquid, and then when the bubble bursts, it spews out these aerosols, right? And then those aerosols were swept up and collected, as opposed to if you just took a glass slide and pipetted a nice droplet and then slowly let it dry over time. And so there was, so here's the fraction of cells that remained active after being dried by aerosolation versus on a surface, and significantly higher survivability if it was aerosolized as opposed to dried on the surface, such a difference that they actually had to blow this up such that you could see this 0.1% survival rate when being dried on a slide. And so more cells remain viable when aerosolized. And there was some also additional studies that looked at high salt concentration actually increased survivability. And so this is to say that instantaneous drying appears to improve survivability. And then I'm on a bit of a side mission to get the word vacuophile into the vernacular here. And so this is actually a really interesting study that showed actual improved growth under low atmosphere, no oxygen conditions for some isolates from Siberian permafrost. And so what this study did is took permafrost isolates and then plated them out either under oxygen atmosphere at our atmospheric pressures, under a CO2 atmosphere, and then under a CO2 atmosphere with 7 millibars of atmosphere. And what they saw is, in this case, you can see for this specific colony here, so here it is not particularly enjoying the oxygen atmosphere. Here it is much preferring the CO2, and here it is even bigger when it actually has CO2 plus lower atmospheric pressure. And then you can also see that in the actual growth of these different colonies over about a month. And so they chose 7 millibars because that's about the pressure of Mars, but it equates to about 30 kilometers-ish in the stratosphere on Earth. So this would be a microbe that actually prefers the atmosphere in the stratosphere as opposed to the atmosphere at the surface of the Earth. And then I also want to point out that aerobiology is not a very novel endeavor in its own right. And if you dig back into the literature from the 1930s, Amelia Earhart was actually doing an aerobiology expedition. And so here's a paper from 1937 lamenting the loss of her plane and actually this global aerobiology collection that she had on board as well. And so the way that they were doing this is with this air hook device. And so it was essentially a glass slide covered in petroleum that was sealed into a tube, and then when the pilot was over the area they wanted to sample, they could open the tube, expose it to the atmosphere, particles would collect, and then when they were done sampling that location, they could close the tube, it would stay sealed until it was returned to a microbiology lab. And so even from this type of study, they were able to see, so this reads, some of the conspicuous objects found on the slide, and they could see, say, stark differences between when they were flying over the Arctic Ocean as opposed to flying over land masses. And so diatoms appearing when they were over the ocean as opposed to pollen appearing over land. So aerobiology's been going on for a long time, and we're just hoping to contribute some of our novel methods to this endeavor. Okay, so actually getting to our project. So here is our overarching question. So our team is led by Jackie Gordial at Guelph. And so, as mentioned, is the atmosphere a living, breathing ecosystem? And then we've divided this up into 3 interlocking sub-questions. And so question 1 is led by Chris Greening at Monash University, and this is getting at the question of are atmospheric communities structured and adapted? Some of the things I hinted to in some of the introductory material. And then I'm leading the metabolically active question. So I mentioned that idea of we know carbon's there, we know they can metabolize these things, but actually are they doing it in situ? That's what we're trying to tackle at Arizona State. And then James Bradley at Marseille, he's looking at the modeling side of things and extending it out to what we might be able to say about exoplanetary atmospheres as well. And so we're sort of wrapping up year 2-ish of our grant, and we spent our initial time on calibration. And so the reason why that's particularly important, so here's actually a recent review paper that came out from our team, and one of the biggest issues we have is contamination control. Because, I don't know, so like the surface of the ocean, maybe that's like 10 to the 6th-ish, 10 to the 8th if you're really got a lot going on, cells per milliliter. Well, in the atmosphere, it's about 10 to the 5th, 10 to the 6th-ish cells per meter cubed. So, you know, we can filter a lot of it, but the density is very low. So we really have to worry about potential for contamination control, because I'm currently spewing lots of microbes just as I'm talking right here, right? And so optimizing for low biomass samples and needing to standardize. So we actually got together in northern Arizona last summer and went over strict contamination control. So this is us in our clean room suits out in the environment, because we're trying not to contaminate what's in front of us, setting up one of these filters on the system, so agreeing upon how to do that. And then we also need to divide and conquer in order to sample. We're trying to do a global sampling campaign, right? So not only do we have to have these protocols, but we have to agree how we're all going to do them the same way, so actually we can parallelize our processing. And so we also unified our protocols, you know, agreed what types of, you know, surface samples we were going to collect in order to compare, and, you know, we're going to collect them from cardinal directions and things like that. So we got all of that under control last summer, and then started going off to our separate environments. So here's some examples of the places we've worked. So we have a lot of members of our team are very focused in polar regions, as well as other marine and dryland systems. And so thus far, so here's a list of all of the ecoregions of the world, and we've sampled almost all ecoregions at least once. That's everything in green, and working on filling in those few black ones. And about half of these regions we've covered by at least 3 samples. And so we're also working on redundancy in our database and sampling different ecoregions multiple times as well. Okay, so then for the last part of this, I'm going to talk a little bit more about the activity measurement side that my lab's been focusing on. So we developed this HERA instrument, which stands for High Elevation Research in Aerobiology platform. And so the reason that we needed to actually do some instrument development is because we needed 2 things. So we needed aerosol filtration, but we needed it at actually low rates that optimize microscopy. So most of the commercial off-the-shelf air filtration things you could buy maximize the amount of particles you're going to collect, and it's a very fibrous, webby system. And so we need a nice, clean, separated sample collection device. And then we also needed to be able to provide gaseous stable isotopes in order to track the in situ activity. So I'll talk about stable isotoping a little bit more in detail, but we needed something that could do both of these things. And so this was all custom designed and machined at ASU. And so the basic idea is just to have 2 filter cassettes and then a custom manifold that allows us to switch between different operations. And then the cassette interior shown here, designed to hold 0.2 micron polycarbonate filters. And so the way this works is we have essentially 2 different modes. So we have one mode in which it's open to the outside environment, and then we pull down through here, and then filter air equally onto these 2 different filters. So this would be our sample collection phase. And then we can move on to an incubation phase, where so this valve closes, this valve opens, we can deliver our labeled isotope gas, and it exposes the filters for the length of our incubation phase. And then we can return back to the initial position and vent the isotope label for the sample to then be returned. And so some of our initial testing here, so this is just kind of a quick overview, but just with cyber DNA staining microscopy, you can see that northern Arizona samples look different than Sonoran desert, like southern Arizona samples. And so we're seeing, distinct morphologies on our samples. And here's actually the HERO platform on NSF Neon Tower up, multiple meters above the ground there. And so the idea of having two filters is we can actually send filters through different pipelines. And so one will be doing microscopy and DNA staining and imaging, like I just mentioned, to get a sense of the abundance. And then the other arm of the pipeline is the stable isotope probing side. So we can take it and do SEM EDS analysis to look at morphology and elemental composition, and then finally take it onto the nanosims, which gives you single cell uptake information. So to talk about this in a little more detail, so this slide is basically sort of the type of work that my lab does. And so the basic idea is if you take a cell and you break it down into a chemical formula, it's going to be some combination of carbon, oxygen, hydrogen, nitrogen, and phosphorus. And those are your options for any sort of labeling in order to look at anabolism or growth, right? The idea is you are what you eat. So if you eat a stable isotope tracer, then you're going to take that up into your biomass and be able to trace it around. And so just to walk through how this gets us to the nanosims, deuterium is a great option because it's not very abundant. This is its natural abundance here, 0.015%. So I'm indicating that by this tiny yellow dot on the cell. So what we can do is we can create experimental conditions that are artificially much higher in deuterium, right? And so if I grow this cell in an incubation condition that is 10% deuterium, say deuterated water, vapor in the case of these experiments, then anything that grows in that condition will take up that label and build it into its biomass. And then you have a range of options. So you could look at the total isotope concentration of all the cells with some sort of elemental analysis, or you could piece stuff out. Right? So you could look at just the enriched DNA or just the enriched lipids or just the enriched proteins if you want to get the additional diagnostic information you would have from those techniques. And then, as I hinted to, we're looking at the stable isotope concentration of single cells. And so what nanosims allows you to do is you can actually map out where the cells are and which ones took up the label. And so this is what this actually looks like. So here's an example with 5 different cells that are all showing in the hydrogen ion image. And then you can see 2 of the cells are lighting up in the deuterium image. So this is an indication of out of these 5 cells that we've imaged, 2 of them were active during the incubation because they took up that deuterium label. So that's the type of data that we're going to be looking at. And here it is. So here's the first attempt at doing this with this apparatus I just talked about. And so we have our cells here, 2 different particles. We found them on the SEM and show that they are carbon-bearing, and then we show that they also were labeled to deuterium. So this might be the potential first evidence of in situ activity from an atmosphere-collected sample. All right, and then I just want to finish up by talking about another arm of the project that's developed that's taken us a little bit more in an arts and humanities collaboration direction. And so this is part of a larger language of landscapes project that's working to bring communities together around ideas of climate change. And they've mostly been working with community groups. And so we're sort of providing a whole, we're doing field recordings at all of our different sites to add that to their sort of audio collective. And so one of the things we do is we sit down and we make what I'm calling a sound map. So here's this one I drew. So this is me sitting here listening for 15 minutes, and then I guess I'm internalizing my internal fruit bat and just trying to think about where all these different sounds are coming from and what I'm sort of paying attention to auditorily. And so we set up these field recorders and listen to those field recordings. So I have a couple of those for you. So I want you to think about which source would generate more microbes, which source has a higher chance of long-term activity in the atmosphere, and then what human-induced sounds do you hear as I play these two recordings? Okay, so the last one was fairly quiet. And so what if I told you, all right, so you have your preconceived things from sound. So what if I tell you this is what the first one looks like and this is what the second one looks like? Would any of you change your opinions about which one appears to be more active or which one appears to be more habitable now that you have visual information? You can think about that. So now that you can see these sites, you now have your auditory information, your visual information, and we can compile that together. And here's some initial takeaways. So the source that would generate more microbes, canonically the literature would suggest, probably A, but it actually depends on which way the wind blows. So the ocean is thought to have about an order of magnitude less cells than terrestrial. So if the wind's coming from the landward side versus the seaward side, it would depend, as compared to a dryland ecosystem. However, as we mentioned, the dryland ecosystem would probably have longer chance of activity, right? Because they're pre-adapted to radiation stress, high degree of temperature stress, and low water activity. So probably B because of its adaptations. And then I think one thing that came out of this that's really interesting is in the auditory landscape feels much more contaminated by these air filtration devices and towers that we're working at. And so you heard all those hums. Those are actually the air towers, right? And so the sampling towers are really loud. And so the audio draws more attention to how they are altering the landscape than if you just happen to just see them, standing there stoically in their metallic nature. So, onward and upward, I'd like to thank everyone else involved with this team, and I'll just particularly give a shout out to John Limas-Alumis, who developed all of the HERA stuff, and with that, I'd like to take questions. Thank you.
Thank you. Great talk. Just a quick question. I have some experience with flask sampling on towers. Do you guys still look for volunteers for the global sample?
I was actually hoping to go the other direction and say if any of you have field sites where you would like us to come sample, we would happily come visit you. So we can maybe figure something out.
Yeah. Well, I could negotiate a deal with Pavel over coffee for a 2-year sabbatical from HFSP.
Okay, I'm going up for sabbatical next year, so let me know.
Questions? Do I get the sabbatical?
Elizabeth, thank you for this excursion. I've spent 35 years as a mathematical physicist chemist studying the atmosphere. Everything from the organic, anorganic, aerosols, isoprene, connection with the CNN. We never ever, with hundreds of colleagues across the world, thousands of colleagues, including here at NASA, look at it from this perspective. Never ever. Never ever. which of course is fascinating because now you look into the biology part of the atmosphere without really wondering about fluxes and measurements and et cetera, et cetera. I mean, we use gas analyzers, we flew them on the aircraft, we were everywhere, but we never came to this point. So my question is now, how bloody hell to connect these communities? Because they may be something which can really move the atmospheric science, the climate science, beyond the current state of the art. So where would you go with your knowledge, with your community, to connect to us, to connect to thousands of atmospheric chemists, mathematicians, having tried to understand the system, but from very different angle?
Yeah, thank you. So I think, one thing that is very exciting to us is that there are, these vast databases that have fantastic information about how packages of air move, right? So we don't have to spend much time on that side of things, and actually, we can go to these neon tower sites where people have huge, stores of data that's publicly available that we can just tap into. So the metadata side of things is fantastic, and so I think where I'm most interested in going is this device. The reason it's kind of small is we actually want to be able to put it on a stratospheric balloon. And so I think we're also kind of at this point where the engineering side is really interesting because it would be nice to follow like a package of air and see how a package of air moves, and microorganisms might if they're adapting or evolving or changing as they're transiting through the atmosphere. So I think it's kind of getting that spatial resolution at the microbial scale is sort of the place that I'm most interested in going. So yeah, it takes a team of everybody. I think it takes everyone to answer this question. So I totally agree that it's great to be at a point where we can combine efforts. Yeah, thanks.
One more question, yeah?
Yes, thank you. This is really interesting work. I just have a question about how variable you would expect this microbiome to be at a given site. You already touched on it with the voice recordings, and now in this last response. I would expect wind, temperature, seasonality, all of these things are going to change not only the microbiome itself in its standard position, but also how much you might expect to come from the earth or the other sources around. So how much of this is known, without going global, just in one particular location, how this changes over time?
Yeah, so the main issue that we are, tackling first, like I said, was this contamination control side of things, right? And so a lot of us have experience in the deep subsurface, which is another place where there's really low cell abundance, really low activity. And so the hope is to take what we've learned there and apply it to the atmosphere, because our assumption is it's kind of a winnowing process, similar to the deep subsurface, right? With time, you're going to just lose volume of cells. But the thing that I think is this open question is will adaptation happen? So with the deep subsurface, the problem is there's not enough energy to divide to adapt, right? But the thing that I think is more possible in the atmosphere is you do have much more of a chance of everything is everywhere and the environment selects is a very common environmental microbiology term. And so there's much more mixing and potential for like global transport at faster timescales. And so I think you might have a better chance of that adaptation, pre-adaptation, allowing longer term survival in the atmosphere than you would in the deep subsurface. So I think initial sequencing is like underway now, and so hopefully we'll have a better idea of that, but these are very good basic ecological theory questions that we're hoping to soon be able to even just start asking with our data set, but I don't have any answers for you yet.
Okay.
Thanks.
2 quick questions still. Last 2.
Thanks, Elizabeth. Really inspiring talk. I was just curious to maybe gauge a little bit further, what would you consider to be sort of like the key result that you guys are going after that would allow you to establish the metabolic activity? Because in the case of D2O, I mean, it's a membrane permeable molecule, right? So it's possible that it could just diffuse into the membrane and then it's inside the cell, but not necessarily being metabolized. I'm sort of curious how you're thinking about that.
A lot. So with the idea is, so I've used a lot of D2O labeling in experiments before, and what we have come up with is just extremely long washing protocols, right? So like allow time for anything that diffused in to diffuse back out again. But the thing that's very interesting and challenging about these samples is we're trying not to add water. And so what we're struggling with right now is when is it worth adding water? So as long as we kill everything before we add water, we know it's at least not affecting the ability for, nothing like wakes up and adds more label, right? But then we lose a lot of the spatial information. Like if it was attached to a salt grain and we add water, we've now lost that salt grain. And so we're working on sort of like parallel instrumentation with the potential for some freeze drying to hopefully see if we can be above background. But worst comes to worst, we watch everything, it's fine. But we're really trying to push the edge of like how long can we go without adding any water at all? Because I think it's so incredibly important scientifically to know what happens with keeping them as close to their in situ condition as possible. So yes. We're trying.
Okay.
And if it takes us 2 more years, now you know why.
Nicola, last question.
So thanks very much. It was a lot of fun hearing that talk. I was just wondering, how important is something like dust and substrates in the atmosphere? And have you got any data on that?
Yeah, so, well, dust and dust storms are a daily occurrence almost in Arizona, and so it's something we think a lot about, often from like a human health hazard, right? Because not just breathing it, but it can contain, viruses and things like that. And so, but the thing that's nice, or I guess another aspect of dust is it provides a potential particle transport source, because there's a lot of theory that suggests the larger a particle is that a cell can like hitch a ride on, it might have a better chance of surviving because it has some nutrient source, it can have some protectant, et cetera. And so dust is really interesting from, it's easier to trace, we kind of know where it's going, and it might provide some sort of like food and protection along the way. So yeah, dust is a really interesting phenomenon to think about in the context of this, yeah. And I also add, we're doing a lot of wildfire sampling as well, and so it's sort of thinking about particles in a kind of slightly more human-induced way too. Yeah, thanks.
Thank you. Thanks, Elizabeth. Great talk. Thank you very much.